Thank you for joining us at the 5th Annual Clinical Diagnostics and Research Online Conference. Life Technologies brand of Thermo Fisher is a proud sponsor of today's webinar on sequencing gene fusions. My name is Reena Karunaratne, and I'm a marketing manager at Life Technologies and will be your moderator for today's presentation. I'm proud to introduce Kelli Bramlett, who is our first speaker in today's presentation. Ms. Bramlett leads the RNA sequencing applications team at Life Sciences Solutions. Prior to joining Thermo Fisher Scientific, Ms. Bramlett was a drug development scientist at Eli Lilly and Company, where she led a team of scientists specializing in gene regulation and nuclear receptor biology to identify novel drug targets. I'm also proud to introduce Jeoffrey Schageman, who will be our second speaker in today's presentation. Jeoffrey Schageman is a staff scientist in the RNA sequencing applications team at Life Sciences Solutions, Thermo Fisher Scientific. He specializes in the development and implementation of analytical methods for NGS and gene expression analysis. Kelli and Jeoffrey will be speaking today on multiplexed fusion gene detection using next-generation sequencing. Also, at the end of the presentation, I will be facilitating Q&A, so please feel free to submit questions during the presentation on your screen. And now I would like to turn it over to Kelli and Jeoffrey for their presentation.
Hello, everyone. It's morning in Texas, which is where I am, so I'm going to say good morning. And thank you for devoting a little bit of time today to learn about a new technology we have to detect fusion genes in a highly multiplexed fashion using next-generation sequencing. Today, I'm just going to talk a little bit about some of the challenges with fusion transcript or fusion gene analysis currently. Then I'm going to tell you about the Ion AmpliSeqRNA technology that is a new technology recently introduced. A little bit about RNA isolation from particularly difficult sample types, and then really kind of wrap these discussions together in a section on using the Ion AmpliSeqRNA technology to detect fusion genes in a multiplexed fashion. Then Jeff's going to take over and talk about specific design of assays to detect fusion genes and also some of the analysis capabilities we have. As Reena pointed out, please, as you have questions throughout the presentation, capture them and we'll address those at the end in a Q&A section. So as many of you on the phone are much more aware than myself, I'm sure, chromosomal translocations are common in cancer. These are rearrangements of the chromosome that often result in a novel gene that is a result of a fusion of two different genes. So we call these fusion genes. And very often, they may produce oncogenic proteins that drive disease progression. These types of events are observed in many types of disease, including hematological diseases, prostate cancer, sarcomas, and lung. Some of the gene families most well understood and involved in these different types of diseases are shown on this slide. BCR-ABL, many people are well aware of in hematological cancers. Also, EML4 ALK fusions, or really any fusion with ALK, has been an area of interest lately for lung. As we think about fusions and how they occur, fusion genes and how they occur and what they're driving, you often find partners that are involved in multiple isoforms of a fusion fusion gene event in various types of cancer. So we would like to be able to detect not only the fusion genes that are most prevalent, but really any events, multiple events that might happen between the driver fusion gene. For lung, this might be ALK, for instance. These fusion genes have been used as indicators of not only cancer type, but progression, and perhaps for drug targets. Detection methods are challenging, though. Currently, really the gold standard for translocation or fusion gene detection is looking at the DNA level using fluorescent in situ hybridization, or FISH. These are DNA probes that are either break-apart probes or breakaway probes, or you may have probes to each member of the fusion gene with fluorescence that you need to see in close proximity to each other. This technology works well, but it is somewhat subjective and requires expert histopathologists to review each slide, and usually multiple pathologists to review each slide. Also, you can really only look at one or two fusion events at a time due to probes and just the procedure of exposing a slide or a patient sample to these probes. Immunohistochemistry is also used to look at the expression of some fusion genes. Again, labor intensive and can really only look at a few genes at a time. Another method that's becoming more utilized is RTPCR. These can be very specific tests for a specific fusion gene event. Can be multiplexed a little bit, but in order to increase the plexy or test for multiple fusions at once, you really start needing more and more sample, which oftentimes is a problem. Fusion genes can also be identified via RNA sequencing, so deep RNA sequencing. This, however, is a very time-consuming and labor-intensive technology that really requires expert bioinformatic analysis that not every lab has, and certainly many clinical labs don't have. So there are some challenges with detecting fusion genes in the field currently. So if we start thinking about what we need to study fusion transcripts or fusion genes, what would be nice? We would like to be able to look at multiple fusions simultaneously, tens to hundreds perhaps. We would like to be able to do this from low sample inputs, so that a fine needle aspirate or an FFPE block or a biopsy sample would be enough to detect multiple fusions at once. And then we would like, comparable or better accuracy than FISH. It would be nice if it was easier to perform than FISH, a fast time to results, and potentially a lower cost, because you would look at all fusions simultaneously. To assay them individually now starts incurring quite a bit of cost. So we'd like to be able to address all of these key factors. So in comes a new technology, Ion AmpliSeqRNA. This is a method to sequence RNA in a targeted fashion. So instead of creating a library of all the RNA in a tissue or a sample, this technology allows you to specifically look at individual RNAs that are of most interest to you. So it's very highly multiplexed targeted RNA sequencing. We can look at gene expression measurements and get nucleotide resolution for those specific amplicons that we're going to use during the Ion AmpliSeq technology. We can look at from 12 to 1,200 genes at once, and importantly, you only need about 10 nanograms of compromised RNA or RNA from preserved tissues, such as formalin-fixed, paraffin-embedded tissues, denoted here and further in this presentation as FFPE. We would also like to be able to barcode multiple libraries and sequence them all at once, further driving down costs and maximizing time and turnaround time. Additionally, we would really like an easy and simple data readout that didn't need, a large group of specialists or bioinformatic personnel to interpret. So we feel that we've accomplished this with Ion AmpliSeq targeted RNA sequencing. What this is basically gene-specific PCR in a multiplexed fashion that we then sequence. So we sequence PCR amplicons. And the way I like to think about it and describe it is shown here on this slide. Many of us are familiar with TaqMan, or quantitative RTPCR, and the way that it works. You have a gene-specific forward primer and reverse primer with a gene-specific probe that sits down on the amplicon that's generated by your forward and reverse primers. The abundance of the gene is then measured by reading the fluorescence signal that you get from the probe, the TaqMan probe. That's shown at the top of the slide. At the bottom of the slide, we're showing the correlation with the TaqMan technology and Ion AmpliSeq RNA. Similarly, we have gene-specific probes, primers, forward and reverse primers. Importantly, we can put up to 2,400 primers or 1,200 assays in one amplification reaction. And instead of needing gene-specific probes and fluorescent signals to read the abundance of each of the genes interrogated, we simply amplify the specific genes and then add adapters that are specific to Ion Torrent sequencing platform. So we actually sequence through the PCR amplicons that are gene-specific, and we read the actual sequence. The amount of sequencing reads that map to each of your gene-specific amplicons correlates to the expression level of the gene interrogated. So this is a very creative way to utilize next-generation sequencing to specifically look at up to 1,200 genes that are of most interest to you with very low input of RNA. This is what the workflow looks like. Again, you only need 10 nanograms of FFPE, and that can be highly degraded FFPE RNA. So this is an important factor that we actually developed into this technology. We wanted the Ion AmpliSeq RNA sequencing capability to be compatible with FFPE RNA or other compromised RNA. So we've designed assays to be fairly short so that the amplicon generated has a high chance of being available in highly degraded and compromised FFPE RNA. So looking at the workflow on the left-hand side in pictorial representation, on the right-hand side in words, we start with your total RNA and have a reverse transcription step. that is randomly reverse transcribed your your RNA into cDNA. The cDNA is then subjected to gene-specific amplification of, again, up to 1,200 targets in one amplification reaction. We then go through a digestion that removes the primers so that you don't waste sequencing space, sequencing through the known sequence. And we add on then through a ligation ion torrent specific adapter sequences. These can either be plain ion adapter sequences, or they can be bar-coded. We always utilize bar-coded adapters, and that allows you to just add a molecular barcode identifier to each library. In this way, you can multiplex or pool multiple libraries together for template prep and sequencing so that you really maximize your sequencing space. And then you create at the end a sequence-ready, targeted, RNA-derived library. The whole workflow takes about 5 hours. Very general molecular biology techniques, all bead-based and requiring no specific instrumentation that a molecular biology laboratory, capable laboratory, wouldn't have. So, as I said, we can multiplex up to 1,200 genes in one amplification reaction, and that's what I'm showing here. The Ion AmpliSeqRNA technology is shown on the y-axis here, and we're comparing that to TACMan. So that's TACMan data is represented on the x-axis here, and importantly, the about 1,000 gene expression assays shown here were accomplished in one multiplexed PCR amplification for Ion AmpliSeq, and for TACMan they were all done individually in 384 well plate format, requiring much more input RNA. So what these dots represent is the expression level of each gene that is interrogated in this panel, as determined by TACMan using fluorescence to estimate the expression level of each gene on the x-axis, or on the y-axis using sequencing reads to estimate the expression level of each gene. As you can see here, the data you get out is very comparable. So the sequencing technology data is very comparable to quantitative PCR data. In this case, we're looking at expression of these 1,000 genes in the highly assayed and well understood control RNAs of human brain reference, which is abbreviated here HBR, and the universal human reference RNA, which is abbreviated here UHR. And we're looking at fold changes of the genes between those two RNAs. This is really just to mimic a type of experiment that may be run either in a laboratory or a translational research or clinical setting, where you would have maybe a control sample and your sample of interest, and you're looking at the difference between these genes and expression levels between your two RNAs. So hopefully I've convinced you that this new technique is exciting and a great way to look specifically at the RNA expression that might be of most interest to you in your studies. One very exciting thing that you can do with this technology is go to AmpliSeq.com and build your own gene expression panel. So if you have a list of between 12 and 1,200 genes that are interesting to the biology or the disease that you're most interested in, go to AmpliSeq.com and build your own panel. You basically log in and submit your gene list, and very quickly you get an email back saying your panel is ready, and you can go look at it and review your panel to make sure that it's going to meet your needs before you order it. We expect you to get your specific panel in about 10 days, and the gene expression panel would be utilized with the Ion RNA AmpliSeq library kit. On the next few slides, I'll just give you some screenshots of what it looks like at AmpliSeq.com when you go in. You can start a new design and you select RNA, focus design as seen on the left-hand side of your screen here. And automatically it'll select human for you. Right now there are only human designs available at AmpliSeq.com. And then you get a screen that says add your genes of interest. You can either do that individually in the dialog box shown here, or you can upload a file. You then hit submit, and within about, I would say, 5 to 10 minutes, you get an email back saying you have a new design ready at AmpliSeq.com. When you go to your AmpliSeq.com account and review your design, you're able to look specifically at the genes the designs that have been suggested for you. It's hard to see details from this screen, so I have a little bit of automation that will allow us to look specifically at some of these sections. Your designs come back and are assigned to various sections. This yellow one at the top says needs attention. And what that might be, I've blown it up a little bit in the screen here, is maybe letting you know that you've selected a gene that is very highly expressed. Sometimes people are used to using highly expressed housekeeping genes for normalization of data. If you're using quantitative PCR methods, that's really a recommendation. We have different recommendations for this RNA AmpliSeq technology. We normalize the data only by the total number of reads to a library, so you don't have to use an endogenous gene normalizer to get meaningful expression results out of your sequencing data. So we don't recommend including genes like GAPDH or beta-actin or 18S that you might have used in the past with other technologies. That is mostly because they're so highly expressed, they soak up a lot of sequencing reads and may actually decrease the sensitivity of your design to the genes that you're most interested in. So what we have is a little dialog box that pops up and says, this gene you've selected is highly expressed and may interfere with your panel. We have some alternative suggestions for you. So that's something that might pop up in a needs attention section of your design. In the green section are things that are designed, and you can select one of your genes and expand the information by toggling on the caret next to a specific gene, and it'll give you information about all the transcripts that are expected to be detected by the assay that is presented to you. So this may be useful information for you. You can also link out to these through a UCSC browser and visualize the different transcripts assigned to a gene and where our primers will sit down and just make sure they're going to work for you before you order anything. After you order and get your custom panel, run your reactions, make a library sequence, you then are able to analyze your data through a simple plugin on the Torrent Suite server. So this will happen basically right on the sequencing instrument and the computer that runs the instrument. It's called a coverage analysis plugin, and it gives you some nice high-level information about your run. the number of mapped reads, as shown here in the middle of this report, and the percent of reads that were on target. So we generally see very high percentage of our reads, more than 90%, that are on target, and meaning the sequencing read maps to one of the amplicons in your panel. We also have some metrics down here about the number of amplicons in your panel, and how many of the genes or amplicons in your panel are detected at 1 read, 10 reads, up to 100,000 reads. So what this kind of gives you a quick snapshot of is the varying expression level of the RNAs in your sample that you interrogated with your panel. So many of us are used to thinking about DNA, where you have expected numbers of genes at the DNA level. You expect 2 copies. In RNA, because of the nature of RNA, you may have vastly different numbers of RNA transcripts present for a specific gene. So what we target here with RNA AmpliSeq is being able to detect genes that are present at very low levels in the same amplification reaction as genes that are present at tens to 100,000 times higher levels than the most lowly expressed gene in your RNA. So we can cover about 5 logs of dynamic range with this technology, which really allows you to focus all your sequencing effort and throughput on the genes that are of most interest to you. Importantly, the coverage analysis plugin also allows you to export the number of reads that are assigned to each amplicon or each design in your panel. And this can be imported into Excel or some other analysis package or statistical package that you may use, so that you can compare libraries or look in more detail at the sequencing results. Again, we just use simple reads per million normalization, or really normalized by the total number of reads to each library. So it's a very simple normalization that you need here. So hopefully I've introduced an interesting new technology to you, and if you start thinking, we've been talking about this in terms of gene expression and looking at up to 1,200 different genes at the same time. So if we go back to our initial discussion about fusion gene detection, you can start thinking about unique ways to use this new technology, perhaps, to interrogate multiple gene fusions at once. So what we'd really like to do is look at multiple fusion events from one very small sample, whether it's a blood sample, cells from a biopsy, or a curl from an FFPE block. Very little nucleic acid amount that we would like to interrogate all of the fusions that might be of interest to your disease state at once. And again, we'd like it to be competitive and offer advantages over the gold standard of FISH. So if you combine the RNA AmpliSeq technology I've just talked about with some unique RNA purification capabilities that Ambion has just been a real champion of throughout the existence of the company, we can really start building a sample to sequencing solution here. So at Ambion, we have a lot of expertise and history in isolating RNA from all sorts of tissues. Of course, you always want to use the highest quality RNA you can to measure gene expression or any type of test you would want to do for RNA. However, we know that oftentimes you don't have pristine RNA. These are real-world samples, very often patient tissues. And so, importantly, AmpliSeq RNA works with even degraded RNA, as I mentioned before. And an example of what degraded RNA might look like is shown here on the right. These are representative of bioanalyzer trays. Normally, if your RNA is very intact, you would see something like the top profile, which has 3 clear peaks on it. But really, we focus on the larger peaks to the kind of middle of this profile. Those represent 18S and 28S ribosomal RNA. Total RNA has a large content of ribosomal RNA. About 80% of total RNA can come from ribosomal RNA species. And that's what's shown at the top profile here. A more common RNA profile that you would get from a tissue that you then try to extract RNA from is shown here in the middle, and we would call that partially degraded. So you can still visualize the 18S and 28S peaks. You see that the 18S peak now has become larger than the 28S peak. That's because those larger 28S ribosomal RNA species have been fragmented. And that's what's contributing to the spiky looking profile to the left of the ribosomal peaks. On the last lower profile here, this is highly degraded RNA. This would be what you would see if you isolated RNA from an FFPE type of sample, or sometimes just from a sample that took too long to get into a preserving buffer, or sat at room temperature for too long. RNA is very labile, and so it degrades rather quickly if you don't protect it well. So this strongly degraded RNA, you can see no more clear ribosomal RNA peaks from 18S or 28S, but this type of RNA still works in the RNA AmpliSeq technology, again, because the assay designs are optimized for fragmented and degraded RNA. So that's one really great thing about AmpliSeq RNA. As you know, FFPE is of interest for not only research but clinical settings as well. This type of preservation or fixation is really widely utilized by pathologists in particular. It's a really nice way to be able to get good histology on a specimen. The bad part about it is that it really compromises your ability to get molecular information, high quality and reliable molecular information out of that same tissue. The formalin fixation process really damages nucleic acids, DNA and RNA. There's chemical adducts and cross-linking that happen between the nucleic acids themselves and nucleic acids and protein, and it just makes it very difficult to get reliable molecular information out of FFPE. However, technologies are advancing, not only in isolating the highest quality RNA and DNA you can get out of FFPE, but also technologies like I've been describing that lend themselves to interrogating highly degraded and difficult chemically modified nucleic acids. So currently quantitative RT-PCR is utilized fairly successfully with FFPE RNA. We're now getting protocols that are working better with deep RNA sequencing, but really Ion AmpliSeq is a great way to look at multiple genes and get lots of information out of a very small amount of compromised nucleic acid from FFPE. Our validated and recommended method to isolate RNA and DNA from FFPE is using the Recover All Total Nucleic Acid Isolation Kit. This has been published as a preferred method for extracting total RNA. Definitely suitable for quantitative RT-PCR, and what we've done in developing the AmpliSeq RNA technology is utilize and test all of our panels with Recover All isolated RNA from FFPE. So this whole workflow has been fully verified and validated in our laboratories here. So hopefully I've talked to you about a couple of different pieces here. Isolation methods to deal with FFPE in particular. I focused on FFPE. We also have recommended isolation, RNA isolation procedures from blood or very few cells. Also exosomes. We've been working on those if those are of interest to you. We've talked about highly multiplexed, targeted next-generation sequencing technologies that allow you to look at multiple genes at once from very low amounts of nucleic acid. And what I'd like to bring it together now to address is combining these technologies to really look at fusion gene detection in clinically relevant sample types. So hoping to get something with better accuracy and better ease of use than current methods like FISH, faster time to results, and a lower cost. So at this point, when we start focusing on how we utilize these technologies to interrogate fusion genes, I'll pass it off to my colleague, Jeff Schageman. Yeah.
Good morning, everyone. For those of you that are still in the morning section of your day, I'm Jeff Schageman. I work closely with Kelli, and we work together developing strategies for not only how do we find the most optimized and best solutions at a reagent level, but also an analytical level. It's not enough just to walk you to the pond and then not allow you to drink, right? So we need to have a full solution. And so what I'm going to concentrate on here is more about once you have your panel for looking at multiple fusion genes at one time, how do you analyze those? So it's not enough just to say, here's your sequences and go. We wouldn't do that. We put together a full solution, and I'm going to talk more about that now. So before I do that, what you're seeing now on the screen is a couple of examples of some very notable publications describing discovered fusion genes and how they can cause deleterious events in metastatic cancers and cause metastatic cancers, and also their basic characterization, whether it be through looking at FISH or quantitative PCR and that kind of thing. So this is just pointing out that we realize there's a need in the field for getting some specificity and ease of use, as Kelli had talked a little bit about before. So how do we actually design an assay to interrogate these fusions in a panel? What you're seeing here on the screen to the left is the basic RNA AmpliSeq workflow. What we want to do is instead of just designing primers to a particular transcript of interest that might be well annotated in the genome, What we'll do is we'll use other metadata or other genomic information that we've extracted through publications or other databases, which I'll talk about in a minute, and then design primers to each of the gene partners, the acceptor, the donors and acceptor genes for that transcript. And the idea would be to amplify across the breakpoint and detect that breakpoint by sequencing. So the figure on the right, you see the 5' gene, the 3' gene, the donor acceptor there. And the lines just below that, those gray lines, are where we want to place the primers and amplify across the breakpoint there that's shown, that green box there with that black line in the middle, that indicates the breakpoint, that breakpoint junction. And so what we want to do is amplify that section with the assay and then sequence the amplicon. The more sequences we have for that breakpoint that we can detect, the more confidence we're going to have that we're actually detecting it. And should mention again, like here, that the level of RNA that this requires is relatively low, even for FFPE. We're talking about 5 to 10 nanograms here still. So I mentioned the sources. So how do we go about designing these assays, and where are we going to get the sequence information to design them, to design the primers, essentially? There's a couple of good places to start. In addition to publications and being formed by experts in the field, there's the COSMIC database, which is a catalog of somatic mutations in cancer. And this is a very well annotated database that doesn't just cover mutations in the form of single nucleotide variants, but also rearrangements, chromosomal rearrangements, and making fusions. So we can comb through this database, extract as much information as possible to do our designs. In addition, NCBI has their nucleotide repository of annotation. So in this case, we're looking at EML4-ALK. This is a commonly found, if it is detected as a fusion in lung cancer. And so we can use this GenBank annotation to go through and find the information we need to synthesize these designs. So a few bullet points about how we go about doing this. So we obtain the source sequence, as I mentioned, from one of these databases, or from a paper, or from expert opinion. We generate some candidate assays, so multiple primer pairs across a given locus, and we're really leveraging what we know about Ion AmpliSeq DNA designs, as well as the DACMan designs that have been around for quite a while now. So then we select based on optimal conditions for these fusions. So we want to be able to guarantee that we can sequence through a given breakpoint. So we have a size range for these amplicons that we want to design, given what we know about the average read length coming off of the ion instrument, albeit a PGM or a proton instrument. So we try to ensure that we can sequence through the breakpoint. We also want to be sure that we don't design primers over known variants, so common SNPs in the genome, single nucleotide variants that could inhibit how that primer hybridizes to that locus. And then we do a final check. There's a QC at the end. When we have highly multiplexed panels, we look for primer-dimer interactions, reciprocal gene interactions, and that's just if you have a panel where you have a donor gene, say it's EML4, and then the donor gene and acceptor gene are actually both EML4, but they're involved in different distinct fusions. You don't want to amplify the wild-type transcript. You want to only amplify the fusion transcript that contains the breakpoint. So we do a check for that. Then we also do an internal priming check to be sure that when we amplify one amplicon in the panel, say maybe it's a expression control that happens to be in the panel with your fusions, you don't want to have another primer designed to another target, use that amplicon as a template. So you run your sequencer on with your panel, and you have taken it all the way through to where you have sequence reads from your Ion PGM sequencer shown up here in the left in this flow diagram. So what you're looking at here is the whole analytical flow of how we analyze fusions, and it's all contained within a software package called Ion Reporter. Ion Reporter 4.2 is going to be the new release to come out on June 27th, and will include support for sequence analysis of fusions with AmpliSeq. So starting up on the left again here, you have your PGM sequencer, which has reads which you then can transfer into the Ion Reporter software. What happens at that point is those read sequences are mapped back to a reference which contains your breakpoint sequences and more than likely expression control genes that you wanted to put in your panel. So you have some reads that map, the majority usually map, and some that don't map. So looking at the branch at the, going from top to bottom here, looking at the mapped reads, you have your reads that map directly to the breakpoint sequences, and we can tally the number of reads that map to those breakpoint sequences to essentially serve as a proxy for detection of a given fusion. There's also a branch to the top right after primary mapping for those that don't map. So you have a cadre of reads that don't map to the reference you expected. There's a few reasons for this. There could be some small degree of off-target amplification causing sequence or resulting in sequences that you didn't expect, or there could be low-quality sequences. What we'll do with these is perform a rescue mapping strategy. We'll take each one of those reads, split them in half, and try to look for the acceptor and donor sequences that we expected to be in the panel. So going straight down from the rescue mapping, we want to confirm those pairs, look for them, make sure that the sequence alignment is actually valid, and perform, again, this counting step where we tally the number of valid pairs that we saw. Occasionally, what will happen, rarely actually, is when we look for the acceptor-donor pairs, we'll find something we didn't expect. So maybe an exon from one partner that we didn't expect partnering with on the other side. Essentially, it's an exon-exon pair that we didn't expect to find in the panel. So there is some degree of detection of non-canonical acceptor-donor pairs that we can pick up with this process. And finally, you post-process the data. The program post-process the data, normalize the data. As Kelli said, that's to the total number of reads per sample, and then you can do a visualization of it. And that's what I'll show here on the next couple of slides. So on the slide here, this is the output from the fusion analysis. This is the table view. So you have a locus. This is the chromosomal locus, the type, basically whether it's a fusion or expression control in this particular example, the genes that are involved with that particular target, the number of reads that have been identified and allocated to that target, and whether it's determined to be present or not. And then as you move to the right, you have more annotations in terms of cosmic or NCBI, and what we refer to as the variant ID, which is very similar to the genes or the exon column I just described. And you can dig into some of these links too to find out more information. So the visualization and normalization view that I mentioned in that flow diagram. So this is another section of Ion Reporter for fusion analysis, and what you can see here are some tabs at the top. You have your samples, workflows, and here this is a fusion analysis heat map. You can see the scale ranging from 0, that red box there, all the way to the right where you have the maximum number of counts detected in these samples. And here in this particular example, we're looking at 6 different samples. And I want to point out is that you can mouse over one of the samples and get more information. I know that might be a little bit hard to see on your screen, but get more information about the sample that you just moused over, so you know what you're looking at. And then I want to point out here, I did mention some expression controls, and these we typically add to our fusion panels so we have a better understanding of how the overall experiment went through as positive controls, and the condition of your RNA. So what you'd expect for these controls, which are not highly expressed genes, they're mid to low level expressors, you should expect to see fairly consistent expression here. So the darker the blue box, the higher expression. And it looks like in this case, for all 6 samples, you have consistent expression across all of them. Also, these samples that you're looking at here are some control samples, so we knew what to expect here. In this case, we are looking at known fusion positive samples. These are EML4-ALK fusions that we know to be detected in a cell line, NCI cell line called HCC2228. So these two variants, or these two isoforms of EML4-ALK are in this sample, and we know about that, and we do detect them successfully. And you can see that from the heat map. Another thing that we can typically add to these panels is assays to interrogate the 3' and 5' end of genes. So if your 3' end is a 3' amplicon is designed, or a 5' amplicon is designed, you can look at the ratio of the total number of reads being allocated to each end to see, as at a broad level, more of a coarse level, do we have an indication that a translocation event occurred, to where we'd only see more of a 3' end of a gene and not in ALK, for example, a driver gene like ALK or ROS or RET, then we see the 5' end. So you see here in this example, you're looking at those red boxes in the heat map. The red boxes indicate the 5' end of the gene and 5' amplicon measurement, and the right next adjacent to that is a blue box indicating that we see expression of the 3' end amplicon. So we do see, we could calculate a ratio between the 5' and 3' end to get some indication that an ALK translocation has occurred in this particular example. One of the things we did in development that was very important is we wanted to have a better understanding of what the sensitivity of this particular approach would be of this technology. So we set up an experiment to where we spiked in this 2228 RNA into normal background of human brain reference RNA, which does not have a fusion in it. So H2228 again has the EML4-ALK fusion present in it. And so what would happen is we titrate in more and more of the H2228 RNA into a background of HBr RNA, and how low can we go? So what you're seeing here is a heat map, again, much like the one you just saw, where across the x-axis at the top you have all the different fusions that were contained, fusions and expression controls at the far right, that were contained within this experiment. So there's a bunch of different EML4-ALK variants. There's some KIF5B RET variants in there, CD64 ROS1, so I'm sorry, CD74 ROS1. And then across the y-axis you have all the different titrations samples. So these are 6 different libraries, so six different sequencing libraries that went on the PGM. As you move from the bottom of the y-axis up, you're increasing the amount you're decreasing the amount of 2228 or fusion positive RNA that's in there. So starting at the very bottom there, you have 100% HBr, so all normal RNA, and there's no detection of that fusion as indicated from the heat map. But as you move up, where you have 100%, next up there, you have 100% 2228, you have a very large, very high level detection of that fusion. In this case, it's EML4 exon 6 and ALK exon 20. Then as you move all the way up that y-axis, you get all the way to 99% HBr and 1% H2228. And we still see a detection of that fusion. So that gave us some confidence that we could readily detect the presence of 1% or even less fusion RNA, even in a background of 99% normal RNA. And this is important because as you select from a tissue sample or from an FFPE curl, you don't always get a pure tumor sample. You get some surrounding cells. And you also have a very precious amount of RNA in many cases, and you don't have very much to work with. So having this level of sensitivity is going to be important going forward for your clinical analysis. So also in the development of this, we worked with some other external scientists, some other customers, and we wanted to find out from real-world samples, so their FFPE samples, could we compare our detection method to what their detection method is? And the state of the art right now is still FISH, as Kelly pointed out. So we were going to work with the group that had not only fish data, so what we're calling known truth, to RTPCR. And then, so in this table here, you've got the lung sample FFPE 17 all the way through 31 there. And then moving across those columns, you have the fish results, our AmpliSeq approach results, and then the RT-PCR results for each of these samples. And anything you're seeing in green is where we agree with fish and RT-PCR from their original data. One thing to point out too, I mean, you might be asking yourself, why put a bunch of different fusions in a single panel? Well, if you, in case you're only interested in measuring a single fusion or a single variant, all I care about is ALK. Well, it might be that you know through fish that you have an ALK translocation or indication of an ALK translocation, but you have no idea what the partner is. So using AmpliSeq RNA to measure this, you can actually see what the partner is. So if you know it's just an ALK translocation, using AmpliSeq you can find out it's EML4, and not only just EML4 on the other side, you can find out which exon is involved in that translocation or that fusion transcript. So as I said before, all of the things that are in green here for the FFPE 17 to 31 are where we agree, but what's interesting here, if you go all the way down to the bottom here, FFPE 30, you see greater than 15% positive cells for fish positive cells for ALK, and then by the other technologies, there were negative by qPCR and negative by using the AmpliSeq technology. So this could be an indication that maybe there's a novel fusion. There's a novel fusion that has yet to be discovered, or that was discovered or detected through fish, or it's a false positive using fish. So, as many of you may know, fish does have the propensity to show false positives, and there's some subjectivity there. But one thing we were excited about when it came to using AmpliSeq to do these measurements is we can really get into specificity of fusion detection, what type of fusion we're detecting, and the level, at more of a quantitative level, less than a qualitative level. So this is, we're reaching the end here, so this is a summary slide. And just to go through some of what Kelly's discussed and what I've discussed. So we talked about fusion transcripts, what they are. They are very important for understanding the nature of metastatic disease, especially tumor types. Current methods to do all of this are still very challenging, and the reason we did that, the reason we started thinking about this technology way back when is because we saw this to be a problem in a way that we could really help out and get to some more specificity and sensitivity into looking at fusions and their detection. Kelly talked about the Ion AmpliSeq RNA technology and how we can highly multiplex multiple genes to a single tube, up to 1,200 genes at one time, with really low RNA requirements. And that's really important, like I said, if you have very precious samples. We're leveraging the Ambion history of really working well and coming up with the best possible solutions for RNA isolation. So we have many kits available for lots of tissues and cells, especially FFPE in the context of today's discussion, as well as blood. And then we can take all of that together, and we can customize this solution to detect fusion transcripts. So we can, as I mentioned, we can detect down to 1% tumor RNA. So we can use less of your precious RNA, and test all your fusions at one time. This is very high resolution, so you know all the variants and the partners, the gene partners that are involved in a fusion transcript. The analysis is essentially pretty much canned for you now. You take your sequence reads, you transfer them, export them to Ion Reporter, and you get a very nice detailed table and visualization. So you can quickly determine which samples are positive for which fusion type. And the results that we've seen so far are definitely comparable to FISH, which is currently the gold standard, and RT-PCR detection as well. So how do we go about, how would you in the audience go about trying to get one of these panels put together for yourself? So there's definitely a mechanism by which you can do this, and so what we recommend is that you would put together some accession numbers or some cosmic IDs of the fusions that you're interested in, or at the very least some sequences that could be extracted or derived through a publication. Identify some endogenous controls so we can be sure that the RNA and that the reactions for the whole system are working properly at a technical level. And then who do you talk to to get this done? So you would contact a field application specialist or your local salesperson to get involved in our white glove program. There's one of these for DNA already. We have one for RNA, not just specific to fusions, but for other applications that go beyond the scope of what we currently offer for RNA AmpliSeq. And what we can do is custom design your fusion transcripts, and we'll work with you closely, and probably Kelly will talk through your needs and address any questions that you have. Certainly go to AmpliSeq.com to get some details, more details about the technology, or even if you just want to play around and design your own phantom panel, I guess. Even if you don't even have to order, you just want to see how the system works. There's details about the kits and the reagents that we sell to get all of this done at lifetech.com. Just do a search in the window, top right, on the Lifetech webpage, and search for AmpliSeq RNA. Thank you very much, and with that, I'll open it up to questions, which Kelly will start out with, and I appreciate your time.
Pass it over
to Kelly real quick.
Okay, the first question is about degradation. Kelly, you touched a bit about sample degradation, and the question is, how can you determine the quality of the samples being tested?
That's very difficult to do. Let's see. That's very difficult to do with, especially with FFPE RNA. Generally, the way that we determine RNA quality is by looking at visualizing the RNA profile on something like the Agilent bioanalyzer instrument. I showed you some profiles of that. Largely for FFPE RNA, it just looks like a degraded mess. So you can use the bioanalyzer to understand what your average or mean RNA size is, that will give you some indication of what you're dealing with. Our RNA AmpliSeq assays are generally designed to be around 100 to 150 nucleotides. So just visualizing your RNA and making sure that you have a reasonable number of fragments that are at least 100 nucleotides in length would be great. But really, what we're focusing on is using some of these control assays that both Jeff and I talked about in a fusion panel in particular. If you have some nice control gene assays in there, it will tell you your RNA was of high enough quality to have efficient reverse transcription occur, efficient PCR amplification occur, etc. So those are really the best ways to try to evaluate your RNA in this type of assay. Have those positive control genes. Make sure you get good amplification of those. We're running out of time a little bit, so I'm actually going to just ask myself the next question, I think, and it might save us a little time. I see there's a nice question about, can RNA fusions be multiplexed with DNA sequencing? So yes, there are DNA AmpliSeq panels as well to look at copy number variation, hotspots, DNA hotspots. You would actually make your libraries independently. both a DNA AmpliSeq library and an RNA AmpliSeq library use the same barcode systems. So you have to just barcode your libraries intelligently so that you can combine your RNA library and DNA library for template prep and sequencing. And then you just, with our analysis tools, you would say barcodes 1 through 4 need to be processed through the DNA hotspot workflow, and barcodes 5 through 8 need to be processed as an RNA fusion panel. That's all the questions I see. Let me pass back to Raina real quick and see.
All right, well, thank you for all of your great questions. I would like to thank Kelli and Jeoffrey for their fantastic presentation today, and to all of the participants for joining. If you'd like to learn more about Ion AmpliSeq technologies and products available through Life Technologies, please visit our Life Technologies booth. It can be accessed from the exhibit hall using the home button on your screen.